Hello, 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 hello. Hello. Hello.
Hello. Hello. Hello. Hello. is some issue with the mic. Hello. Hello. Am I audible, students? Hello. Okay. I guess now you are able to hear now. So that is what I just said. That we'll be discussing some of the important questions from the PUC first year, diagram based questions, and the data that we'll be discussing here. Okay. Okay. Fine. Now, in this class, specifically, we are going to discuss the diagrams and the diagram based questions, whatever the probability of questions can come in tomorrow's NEET exam. So, without delay, we'll be going to the first slide. Okay. From the first chapter, that is the living world. Remember, it's a very, as we are focused mainly on the diagram and table based question. So, we'll be discussing here only with respect to that. Now, remember that in this case, the first table here, the species being the lowest unit of classification. As we go ahead, species, genus, family, order, class, phylum, division, kingdom. Again, if you know, you have to remember that the word phylum, the word phylum is applicable only for the animals, whereas division is applicable only for plant species. Okay, so when we are describing or when we are writing the taxonomy category for plants, we use the word division. If we are planning or if we are writing the taxonomy category for animals, we'll use the word phylum. Okay, so kingdom, phylum, class, order, family, genus, species. Should remember that. And this table, again, the 4 taxonomic categories or classification that is mentioned in your NCERT is these 4. The common name, that's man, housefly, mango, wheat. Okay, 2 from plants and 2 from animals is what is given in your textbook. Now, the scientific name of man is Homo sapiens. Okay, and also you have to remember with respect to the binomial nomenclature. Okay, the correct method of writing a binomial nomenclature or scientific name is very important. Okay, now the first letter should be capital. With respect to genus, the first letter of the species must be small, and if handwritten they must be underlined separately, and the names are derived from Latin, remember that. And if printed, it will be in italics. What does that mean? That means that whenever we are printing in italics form, it is indicating the Latin origin. Remember that. Okay, so the family of man is Hominidae, order is Primata, class Mammalia, phylum. We are using the word phylum because we are talking about animal species. So the phylum is Chordata. Okay, the next species that we are going to discuss is housefly. Common name is housefly. The scientific name is Musca domestica. The genus name is Musca and the species name is Domestica. Okay, it belongs to family Musidae and order Diptera, class Insecta. One very important point about class Insecta is it is the most abundant class in the whole animal kingdom. Okay, and the phylum to which it belongs, Arthropoda. Obviously, if the class is most abundant, if that is belonging to a specific group, so Arthropoda is also the most abundant species in case of animal class of phylum Arthropoda. So that's a very important point that you should remember. Then, if we speak of the next species, that's mango and wheat. Mango, the scientific name of mango is Mangifera indica. It belongs to family Anacardiaceae, order Sapindales. It's a dicotyledone. So the class is dicotyledone, belongs to division Angiosperma. Now we are using the word division because we are talking about plant species, right? Next, wheat. The scientific name of wheat is Triticum aestivum. It belongs to family Poaceae. Very important because Poaceae is an additional family that is added in your NEET syllabus. Next, belongs to order Poales. It belongs to class Monocotyledone and the division Angiosperma. Now, in this also, there's something that you should remember. The most or the few things that you should remember here is the family name. Okay, the family name in case of animals ends with AE. If you observe Musidae, Hominidae, so the family name in case of animals ends with AE, whereas in case of plants, the family name ends with ACA. Okay, if you recall some other families of the plants, which is done in your NCERT or prescribed by NTA for NEET syllabus, is apart from Poaceae and Anacardiaceae. Anacardiaceae is not there, but it is mentioned here. One more is Solanaceae, then Fabaceae. Remember, Fabaceae is itself, known as one more name. The subgroup is Leguminosae. Leguminosae doesn't satisfy the ending word aca. Leguminosae is a subfamily of Fabaceae. Okay. Then, apart from Fabaceae and Solanaceae and Poaceae, there is Asteraceae. Okay. Earlier, the name of Asteraceae was Compositae. Now it is known with the name Asteraceae. Okay. Then Brassicaceae. Now, Brassicaceae was earlier called as Cruciferae. Now it is called as Brassicaceae. Then Poaceae. Earlier Poaceae name was Graminae. Now it is called as Poaceae. Okay. So these are the other families. And one more is Malvaceae, which is also commonly called as hibiscus family. So remember these things. So I hope it is clear in this first line. We'll go for the next one. Here, moving on to the second chapter, biological classification. The table that is mentioned in the second chapter is this table. So here we have to focus on Monera protista. The most important thing here is the major difference between Monera and protista is that these are unicellular prokaryotic organisms. The Monera includes all the bacteria. The sole members of Monera is bacteria, and protista has unicellular organisms. But what is the main difference? The species that comes under protista are eukaryotes, whereas the species that comes under kingdom Monera belongs to prokaryotes. Okay, the nuclear membrane is absent as they are prokaryotes. The nuclear membrane is absent. The cell wall in case of Monera is non-cellulosic, yes, but it is made up of a very important component which we call it as peptidoglycan. The cell wall is made up of peptidoglycan. Peptidoglycan consists of NAM plus NAG. That's NAM and NAG, N-acetyl muramic acid and N-acetyl glucosamine. So these are the important components of peptidoglycan. Then obviously you need their cellular organisms. Mode of nutrition, they are autotrophic. In autotrophic, again they can be photosynthetic or chemosynthetic. Photosynthetic, which can utilize the sunlight and prepare their own food. whereas chemosynthetic, they will break the inorganic substrate, and by utilizing that energy they are going to prepare their own food. They are not only autotrophic but they are also heterotrophic. The most abundant bacteria, remember these, the most abundant bacteria are heterotrophs. Okay, they can be either saprophytic, parasitic. Okay, it's very important here. Then moving on to protista. Now protista are unicellular eukaryotes. The cell wall is present in some, whereas in some they are absent, because in protista we'll discuss in the next slide that there are total of 5 subclasses, in which the initial 3 classes are like plant like, whereas the remaining 2 are animal like. So we'll discuss about that. Nuclear membrane is present because they are eukaryotes. They are also cellular, but initially 2 groups or 2 classes are autotrophic, that is they can prepare their own food by the process of photosynthesis. The later 2, the third one is a kind of mix. It acts as mixotrophic organism because in presence of sunlight it can utilize the sunlight rays, photons, and it can prepare its own food. In absence of sunlight it is going to act as a heterotrophic organism. So that's euglenoids. Okay, they are going to act as autotrophic as well as heterotrophic depending on presence or absence of sunlight. Then the remaining 2, they are obviously they behave as heterotrophic organisms, the slime molds and protozoans, which we'll discuss. Then fungi. The very important point here is cell wall. The fungi is made up of a very important component which we call it as chitin. Okay, remember chitin is not just found in the cell walls of fungi, but it is also found in the exoskeletons of arthropods. So you have to remember this. Okay, a nuclear membrane is present. Almost all the examples in fungi, they are multicellular except one, and that one is yeast. Remember that. Okay, yeast is an example wherein it is unicellular. So the unicellular fungi that we see here is yeast. Apart from yeast, all of or most of the fungi, they are usually multicellular. They are heterotrophic. They are purely heterotrophic. Okay, so the mode of nutrition can be saprophytic or parasitic. Saprophytic means the organisms which feed on dead and decaying matter. Parasitic, which may be a endoparasite or ectoparasite, usually parasite that is responsible for causing of some diseases, which we'll speak. Rust smut is a parasite that we see in case of plants. Okay, plant diseases, smut and rust. Okay, even on mustard, that's a very important point which most of you will miss in the introduction part of fungi. There's something called as the smut disease, the mustard. Okay, mustard, the disease is caused by which of the fungi, that you should remember, and you should not forget that also. That is an example seen in Phycomycetes. Okay, then plantae, obviously they are eukaryotes. The cellulose, the main component of cell wall is cellulose. Cellulose is not the only component that we see in cell wall. There are again different components, hemicellulose, pectin. Okay, so other components are present, but mainly we see cellulose. Then they can be seen at tissue or organ level of organization. They are purely autotrophic, with exception of Cuscuta. A very important example here, which is seen even in case of the second year as well. In second years, there is an example called as Cuscuta, and also mentioned in your plant kingdom. Cuscuta is a heterotrophic plant, the exception that we see here. Okay, Cuscuta is heterotrophic. It derives its nutrition from the hedge plants. Remember, Cuscuta is heterotrophic. Mainly, if you have to say parasitic, okay, it is also an example for parasitism in your second year. So Cuscuta which is a parasitic plant. It is acting as a parasite on hedge plants. Okay, so that's one more example. Then Animalia. In case of kingdom Animalia, they are eukaryotes. The cell wall is absent. That's a very important character. Cell wall in case of animals is completely absent. The outermost component of cell wall is cell membrane, which we also call it as plasma membrane. The body of organization can be seen at tissue, organ, or organ system level of organization. They're also heterotrophic. So initially, if we have to speak about few things about the chapter here, biological classification, the earliest attempt was made by Aristotle. Later on, there was two kingdom classification, followed by 3, 4, and 5. Among 2, 3, and 4 kingdom of classification, the common two kingdoms that were kept constant throughout the classification was Plantae and Animalia. But later on, as the scientists found that the requirement of the classification that was done in classifying the organisms into kingdom Plantae and Animalia was not sufficient. It was lacking somewhere, because here the fungi had cell wall, but they cannot prepare their own food. They are heterotrophic. Then how you can place fungi in Plantae, right? So the fungi was moved from Plantae and made into a separate group, that's kingdom fungi. Remember, kingdom fungi is also referred as a kingdom mycota. Okay, that's one more important point. Then Protista and Monera, they both are unicellular organisms. Okay, whereas the examples, most of the examples of Plantae and Animalia, they are kind of multicellular organisms here. So again, we classified. You could have asked, sir, why there is a need of two kingdoms? We could have placed both of these organisms in just one single kingdom. It is because, again, as we discussed, the different nature of cell, that's prokaryotic and eukaryotic. Therefore, the kingdom Monera was again further classified into, two divisions. If we have to speak, there are total of 6 kingdom classification, which we divide into 3 domains. Kingdom Monera is again further divided into 2 different groups. The first group being Archaebacteria, okay, Archaea, which we call. The second group is Eukarya. Then the third group is the Eubacteria was the second group. Eukarya, under Eukarya, all the remaining 4 phylums will come here. Okay, so if I have to just say you, just a second. The kingdom Monera was split into 2 different divisions. One is Archaebacteria and the other is Eubacteria. And all the remaining 4 kingdoms, that's kingdom Protista, kingdom Fungi, then Animalia, and Plantae and Animalia. Okay, so all these 4 different kingdoms were placed under one more division, that's one more domain, that's Eukarya. So the total there are 3 domains. The first domain being Archaebacteria, that's Archaea. The second domain is Eukarya. The third Eubacteria. The third domain is Eukarya. Eu means true, karya means nucleus. So the organisms which are having true nucleus were placed under one single domain. Okay, next. So yes, this is again one more very important concept here. The different shapes of bacteria, which can be cocci, the spherical shaped structure, bacilli, rod shaped structure, spirilla, spiral shaped structure, vibrio, comma shaped structure. So these are the 4 different shapes of bacteria. And then if you have to speak, filamentous blue-green algae, okay, it's an example for cyanobacteria. Now, in cyanobacteria, there are different examples. nostoc, anavena, and also remember oscillatoria from the second year point of view. So there are total examples that is mentioned in your NCERT for cyanobacteria are these three, that is nostoc, anavena, and oscillatoria. The very important point you should remember here is the it is a filamentous structure, okay, wherein it is forming a chain like structure. Then the cells are covered by mucilaginous sheath, okay. The single cells, they are all covered by mucilaginous sheath. There is a heterocyst. If you see, if you observe, all these cells, they are different from this cell, okay. This is referred as heterocyst. Now, what is its role? What is the role of heterocyst is? It helps in fixing of atmospheric nitrogen, okay. And the remaining other cells, it's also useful in storing the food. So that's one more very important point. Then if you have to speak how the bacteria will divide, bacteria will usually divide by fission, okay. If you see here, a single bacteria is dividing into two different bacteria, bacteria with cell membrane and the genetic component here. As you know, bacteria are prokaryotes. They do not have a membrane bound nucleus. So the nucleus, we are not going to call it as nucleus, but we are going to refer it as nucleoid. Okay. Then moving forward onto the next concept. So again, these are again diagram based questions. First is dinoflagellates. Okay. Remember, there are, as I said, prokaryotes, I mean the Protista, kingdom Protista has four different classes, that is C, D, E, S, P. C stands for chrysophytes, D stands for dinoflagellates, E stands for euglenoids, S stands for slime molds, and P stands for protozoans. Okay, so the chief, okay, the very important, not chief, but chief, very major, okay, components of the oceans are diatoms. Okay, so that is an example for chrysophytes. Remember that, okay, which comes under chrysophytes. Not only that, okay, the golden algae, okay, they are also present in the chrysophytes. Now again, diatomaceous earth is also from chrysophytes, wherein we are going to obtain the diatomaceous earth, which is being present beneath the oceans. How it is formed? As the chrysophytes, the cell wall of chrysophytes is having silica as its component, which exactly fits into a soap box kind of the structure. Now, this silica is not degradable. So as the years progress, this silica will settle at the bottom of the ocean and it forms layer after the layer. That is itself what we call it as diatoms. Okay, just a second. Hello. I guess again voice is not audible. Hello. Yes, the sound is little less audible. Slowly it is audible. Just a second, I'll recorrect it. Hello. is the sound audible? Hello. 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 Am I audible now? I hope I'm audible now. Okay. I guess in the initial part, you were not able to hear. So just increase the volume by little. I guess slightly slow sound has come in the starting, in the beginning. So from now, I guess you'll not be getting any issues. Okay. Now, am I audible? Hello. Yes, students. Okay. Is it audible now? Just once again confirm. Okay. I guess the sound is clear and now we are good to go. So, in the second class, that's the dinoflagellates. Dinoflagellates, you have to remember that the guinolags, example, guinolags Okay, Guanolax is the one which is responsible for causing the red tides, and also they are toxic as well. Now, moving ahead, euglenoids. As we discussed, euglenoids are the mixotrophic organisms wherein in presence of sunlight they are photosynthetic, and in absence of sunlight they are acting as a heterotrophic organisms. Slime molds. Now, slime molds, it's very important because they kind of behave like fungi. One very important point that you should remember here is the protistans. They are primarily aquatic, but the slime molds, what happens is they aggregate themselves and forms a structure which we refer it as plasmodium. Not the plasmodium which you have studied, okay, not that plasmodium, not that, but this aggregation of slime mold is itself called as plasmodium. Okay, and during unfavorable conditions they form the spores, and then those spores will be dispersed further so that again slime molds will regrow. Okay, then the last group, that is protozoans. Protozoans are again subdivided into 4 different groups, that is amoeboid protozoans, ciliated protozoans, flagellated protozoans, and sporozoans. Okay, amoeboid protozoans, they move or they are having locomotor structures, pseudopodia as their main locomotor structures. Ciliated protozoans are the organisms which have cilia present in them. Flagellated protozoans are the protozoans which have flagella present in them and which acts as main locomotory organs. Sporozoans is a class of protozoans wherein they do not have any specific locomotory structure. Okay, these sporozoans, the example for sporozoan being your malarial parasite, that's your plasmodium vivax. Okay, then you have to also remember about sleeping sickness. Okay, that's one more example which you should remember here. So these are all the things that we discussed here. Moving forward to the next one, fungi. Okay, mucor, aspergillus, and agaricus, some examples that is mentioned in your NCERT regarding fungi. So totally in fungi also there are 4 different groups: Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteriomycetes. The Phycomycetes, the example for Phycomycetes being Mucor, Albigo, and Rhizopus, the bread mold Rhizopus, which you should remember, and the only group to be unbranched and aseptate. Okay, that's very unique property about Phycomycetes. Then Ascomycetes, they can reproduce both by sexual as well as asexual spores. Sexual spores being ascospores, and the asexual spores is usually being the conidia, which you should remember. Okay, then moving on to the Basidiomycetes. Basidiomycetes do not show any asexual spores, and the sex organs are absent, but they usually produce the Basidiomycetes. Okay, basidiospores are being produced by Basidiomycetes. Okay, and then the last group is Deuteriomycetes. Deuteriomycetes, they are called as imperfect fungi. Why they are called as imperfect fungi? Because they do not show any sexual cycle. So sexual spores are completely absent in Deuteriomycetes. Okay, you have to remember this. Next, agaricus, the common name is mushroom. Okay, aspergillus, aspergillus niger, if you remember. Okay, acetic acid. Okay, then one more very important point in Basidiomycetes is regarding the rust and smut diseases. Puccinia, okay, is one of the again example which causes the rust and smut diseases. Okay, Ustiliacospores and Puccinia, they are the examples for fungi. Then you also have to remember about one thing about Ascomycetes. Yeast comes in Ascomycetes. Yeast, The scientific name of yeast being Saccharomyces cerevisiae, which is also called as brewer's yeast, baker's yeast, used in the preparation of beverages, your food items such as idli, dosas. Okay, for preparing that we need yeast. Okay, which is the only unicellular organism here, unicellular organism in fungi being yeast. Rest all are multicellular, and this is non-filamentous. Remember, unicellular and non-filamentous. Okay, then not only that, you have to also remember about neurospora, the genetic organism. Okay, the model organism which we call it as neurospora crassa is an example which is extensively used for carrying out genetic works, biochemical and genetic works. So that's one more thing that you should remember here about the different groups of fungi. Then moving to the next one. Yeah, the last tobacco mosaic virus. Okay, TME, capsid, the single units, the single units, they are called as capsomeres, which may be polyhedral or tetrahedral depending on the structure. Okay, the tobacco consists of the tobacco mosaic virus mainly has the RNA. Then bacteriophage, bacteriophage has double stranded DNA, remember that. Okay, this part is called as sheath. The tail fibers, collar, head, inside the head there is presence of DNA. Okay, next, if you have to go for the next one, these are the examples for green algae, volvox, colony. Okay, an example for colonial algae is volvox. You can see the colonies here. Then Eulothrix being this one, brown algae. Very important point about brown algae is presence of holdfast, stife and frond. Holdfast is meant for attachment to the substratum. Stife is connecting frond and holdfast. Frond is referred as photosynthetic organ. Okay, you should remember this. Then there is also red algae. The examples for red algae being porphyra and Polysiphonia. Also remember Gelidium and Gracilarium. Okay, then the Chlorophyceae, class Chlorophyceae, major pigments are chlorophyll A and B. In case of brown algae, the major pigments being chlorophyll A and C and fucoxanthin. In case of red algae or Rhodophyceae, the major pigments are chlorophyll A and D with phycoerythrin, R phycoerythrin. Okay, the main pigment which is responsible for producing red color is R phycoerythrin. Okay, then the cell wall. Okay, the cell wall of the green algae is made up of cellulose. The brown algae is made up of cellulose and algin. Remember, algin is extracted from brown algae, so you can remember like that. Cellulose, pectin and polysulfuracids are, they are the components, cell wall components of red algae. Then flagella number. Okay, 2 to 8 flagella are present. They are all equal in size. Okay, and they are all present in the apical part. For example, if this is the structure here, the flagella can be 2 in number, and this is the apical part. This part is apical part. They can be 2 to 8 in number. The position is apical. In case of brown algae, there is presence of 2 flagella which is unequal in size, and they present laterally. So laterally means if this is the structure, they are present laterally, sideways. One is longer one, the other is very short one. Okay, and in red algae, the complete flagella are absent. Okay, then moving on to the next one. Yes. So if we discuss about bryophytes here, bryophytes, the archegoniophore, it is the stalk of archegonia. So this is the archegonia. The stalk of archegonia is archegoniophore. It is present on the thallus. Okay, the ventral part, okay, of the thallus is having rhizoids. Rhizoids are nothing but root like structures, okay, which are present at the base of thallus, and on top of that you can find gamma cups. These are nothing but, it's a method of asexual reproduction, which the gamma cups, when detaches from the parental thallus, it can grow into a new, complete new gametophyte, okay. Then Anthridiophore is a stalk. It is a stalk of Anthridia. So this is Anthridia, the male sex organ. This Anthridia will produce antherozoids. Antherozoids are the gametes, okay, which are produced in the Anthridia, okay. And the stalk of Anthridia is called as Anthridiophore, okay. And again we see the gamma cups here, okay. These are the examples of Markensia, the thallus of Markensia, okay. Then you have to remember that there are again two groups in bryophytes. One is liverworts, the other is mosses. So liverworts, they are somewhat primitive when compared to the mosses. So these are the mosses. The example here mentioned here is for sphagnum and funaria and sphagnum. So this is the sphagnum here that we see. It's a moss like structure. Now the funaria consists of the leaf like structure, the stem like structure, and uh, the front capsule part is also present. So this part, it is referred as food part, okay, the food part which is embedded in the gametophytic structure. So actually this is the gametophyte, the sporophyte grows here, the food part is embedded, means present inside the gametophyte part, and the extended part, this part is called as setae, and at the top there is presence of capsule. Now capsule has spores, it has spores, remember spores are haploid, okay. This complete part is sporophyte, inside capsule there is presence of spores, okay. Now why it is present inside the gametophyte? Because sporophyte is dependent on gametophyte for its nourishment, development. The sporophyte cannot produce, the sporophyte cannot produce its own food, therefore it is dependent on the gametophyte for the food, okay. You should remember this. Then moving on to the next one, we'll go quickly, little quick, because we are late on time and we have to move forward. Next example for pteridophytes being Selaginella, okay, Equisitum, ferns, and Salvinia. Don't get confused with Salvinia, and one more is the same, okay, Salvinia, but that is coming under angiosperms. We'll discuss that when we come to angiosperms. Don't get confused here, okay. Then Selaginella and Equisitum, they are very important examples for the production of spores, okay. I mean the cones, okay, the cones are produced here. If you see, usually the pteridophytes reproduce by the production of spores, but here in Equisitum we see the presence of cone. And one very important property of pteridophytes is seed retention, okay. That's what we call it as seed habit. Seed habit is seen in case of pteridophytes, okay. That's a very important point in pteridophytes. Next, gymnosperms, okay. This is Cycas, this is Pinus, and that's Ginkgo, okay. It's also a living fossil. Now the major question that can come from this part is regarding Cycas and Pinus. Remember only about Pinus. If you see the structure of Pinus, it is very large, okay. It kind of appears as like big tree, right? So how you have to remember it, as it is big, obviously it is branched. So the branching is seen in Pinus, remember that. Not only branching, but if imagine if this huge kind of tree is present, obviously both male and female reproductive structures, that's male strobili and female strobili, the cones, both male and female cones are present on the same tree, okay. They are present on the same plant, that's Pinus. Now in case of cycas, as you see, it is a small plant. These are not branches, okay? They're not branch. So obviously cycas is unbranched, and if it is a small plant, the male strobili are present in one plant and the female strobili are present in another plant, okay? So that is what you have to remember here, the difference between the two. Just remember about pinus, it's a very huge tree. Obviously if it is huge, then it shows branching. At the same time, it is, having both male and female cones present in the same tree, okay? That's one thing. Clear? I hope it is clear. And some modifications that you should remember in gymnosperms is regarding needle like structure, presence of needle like structure, sunken stomata. They're all the presence of thick cuticle. They're all the characteristics because to save the water loss or to reduce the transpiration, the gymnosperms have undergone certain changes, okay? That's presence of thick cuticle, the reduce the size of the leaf, forming needle like structures. There are some modifications. Not only that, you should also remember about the the roots of these gymnosperms. They are tap roots, okay? They are taproot system. Taproot is present. Now, not just taproot, these roots are in association with bacteria as well as fungi. If these taproots, if they are association with fungi, they forms a symbiotic association with fungi, and that is what we call mycorrhiza. Now, remember, if you remember, I told that fungi is known with one more name, that's mycota. So, mycorrhiza. Mycorrhiza means what? Rhiza means here root. So myco means fungi. The fungi are in association with the roots of this plant. If they are in association with the roots of the plant, they're helping the plants to get atmospheric nitrogen. Not only that, even some of the cyanobacteria, they can form association with these roots of these plants, and that is what we call it as coralloid roots, okay? These are again few things that you should remember from the gymnosperm part. And in case of angiosperm, dicot plant, that's given here, and this is a monocot plant, okay? You should remember these two. Moving forward, I have to also remember Wolffia and the tallest flower, okay? We'll come to the and angiosperms in the later part. Now moving on to animal kingdom. Radial symmetry. What is radial symmetry? An organism can be equally cut or divisible into two equal halves when cut from multiple planes or more than one plane. If you see here in this example, if we cut this organism, if we divide this organism from this plane, it gives two equal halves. If we cut it from this plane, we are again going to get two equal halves. If we cut it from this plane, we can get two equal halves. So if you are getting two equal halves when cut from any plane, that type of symmetry is called as radial symmetry. And if we can get only two equal halves when cut from only one plane, so only one plane if we use to cut the organism into two equal halves, that kind of symmetry is called as bilateral symmetry, okay? Which is observed in most of the higher animals, which we'll discuss, okay? Now if we have to say it here only, radial symmetry is usually seen in case of your Porifera is a primitive phylum. Moving on to the next one, the next phylum being Ctenophora. Ctenophora and Coelenterata. Both of these phylum, they show radial symmetry, and rest all the phylum, they show bilateral symmetry, with the exception of the echinodermates, wherein the adult they show radial symmetry, the larva shows bilateral symmetry. Then moving on to the next one, the germinal layers, they can be either diploblastic or triploblastic. Now if you see here, the outermost layer, just change the marker here, you see this part, So this is what we call outer ectoderm. And if you see this part, you see this part, this is inner endoderm. The middle portion, if you see here, this part, this is not divided. Okay, the cells are present, no doubt the cells are present, but the cells are not differentiated. So undifferentiated mass of cells is what we call mesoglia. Here in the triploblastic animals, here we can clearly see the outer ectoderm, inner endoderm, and the middle endoderm. So the cells are very well differentiated here. You can observe the cells here, whereas you cannot observe the cells here. So this is the major difference that we see, okay, in diploblastic and triploblastic animals. Presence of two layers, presence of two germinal layers is what we call diploblastic animals. Presence of three germinal layers is what we call the triploblastic animals. Okay, triploblastic animals, it begins from the phylum Platyhelminthes. From Platyhelminthes onwards, all the phylums, they are triploblastic in nature. Before Platyhelminthes, that's Porifera, Coelenterata, Ctenophora. Then your these three phylums, they are diploblastic, whereas the remaining Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, Chordata, they all show triploblastic nature or presence of three germinal layers. Now, don't get confused between the germinal layers and the coelom. This is where most of the people will get confused here. Now, coelom is nothing but it's a cavity. If the cavity is present between body wall and gut wall, we say coelom is present. That means cavity is present. Cavity means the space. Okay. If the space is present between body wall and gut wall, it is what we call it as coelomic. If there is no gap, then what is that? That is what we call acoelomate. Now, if you observe the first, we'll go for C1. C is acoelomate. Now, how to identify here? The outer, this is body wall, completely body wall. Now, the body wall is differentiated into ectoderm, endoderm, mesoderm, everything is differentiated. But now, if you observe here, there is no space here. Okay, this is actually gut wall and body wall, which is not having any cavity in the middle. We'll go for the second one. Now here you can find the outermost part, and here in between this outer body wall and gut wall, here there is some presence of some space here. If you observe here, there is some space. Now this space is itself called as cavity. But if you observe this, in the middle there is actually presence of other tissues or organs. So there is not actually sufficient space. So the cavity is kind of present, but in the form of pouches. Pouches means small areas it is covered. Okay, so such kind of coelom, it is called as pseudocoelomate. Pseudo means false, coelom means cavity. So if there is a false cavity, that is what we say it as pseudocoelomic, wherein the coelom is not completely present. Now if you observe in the first part here, the body wall and the gut wall, in between these there is enough a sufficient space, there is enough cavity, which we call it as a coelom. Okay, so enough cavity, enough space is present between body wall and gut wall, so therefore we call it as a coelomic or coelom is present. Now pseudocoelomate is observed in Aschelminthes. So how you should remember, whenever somebody asks a question, he will tell lie. Lie means false. False, pseudo. Okay. So if somebody is saying false, okay, false thing, that is what we call pseudo. Coelom means cavity. So ask. When somebody asks, he will tell lie. That's pseudo. Okay. Then before Aschelminthes, Platyhelminthes, Ctenophora, Coelenterata, Porifera, they're all acoelomate. And after Aschelminthes, that's Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata, they're all coelomic. I hope this is clear. Moving to the next part. This is again, in short, to summarize, kingdom Animalia, which is obviously multicellular. In multicellular also we see cellular level of organization. They are generally asymmetric. Okay, body cavity is absent, seen in phylum Porifera. If tissue or organ system level of organization is observed, the tissue level of organization is seen in Coelenterata and Ctenophora. They are radially symmetrical, and again coelom is absent. If they are bilaterally symmetrical without body cavity, that's acoelomate, is platyhelminthes. Pseudocoelomate, presence of false coelom, is Aschelminthes. Presence of true coelom is seen in these particular phylums. I hope it is clear. The same thing goes with the germinal layers as well. If they are diploblastic or triploblastic, till Porifera, Coelenterata and Ctenophora, they are diploblastic, and platyhelminthes being triploblastic. From platyhelminthes onwards, all are triploblastic. These are examples of Porifera: Cycon, Euspongia and Spongilla. Okay, remember the bath sponge, it appears like a sponge, so the Euspongia is an example for bath sponge, a common name. Okay, remember that. Then next, Coelenterata, there are a few things which you should remember, some unique features of Coelenterata, that is presence of nidoblast cells, which we'll discuss. But apart from that, there is one more very important feature of Coelenterata, is presence of two different forms. One is medusa. This is medusa form, and this is polyp form. The example for medusa being Aurelia. Aurelia, the common name for Aurelia is jellyfish. Okay, and the polyp form is Adamsia. Also we can see Hydra as well. Hydra is also polyp form. Now the major difference is it is free living, wherein it can easily swim. The medusa form can easily swim, whereas the polyp form is found attached to the base of the substratum, and it is not free living. Okay, so this is found attached to the substratum, and this is free living, wherein it can swim easily in the water. Okay, then this is how the nidoblast cells look like. If you see here the tentacles, okay, these tentacles are present. These tentacles, these tentacles are responsible for movement, capturing. Not only that, even they possess the nidoblast cells. They are stinging cells. Okay, these stinging cells, when sting to any of the particular fishes, it may make that fish or species, it has some toxin present in them. The stinging cells, when injected into those particular fishes, they become the food for these organisms. Moving on to the next one, example for Ctenophora, Pleurobacchia. Remember this example, and if you see presence of 8 ciliated rows. Okay, 1 2 3 4 5 6 7 8, 8 ciliated comb plates are present, which is very helpful for the moment. The tentacles are present, and the very important feature of Ctenophora is bioluminescence. Bioluminescence, the ability of emitting the light. Okay, the examples for platyhelminthes is tapeworm and liver fluke. Okay, again one thing that you should again remember in platyhelminthes is most of them, they are endoparasites which are present inside the body of the host. Now Aschelminthes, very important thing, again Aschelminthes, the roundworms which is present. These roundworms or Aschelminthes, the example here is of Ascaris. Remember it from your second year point of view as well, Ascaris lumbricoides. Okay, The practical, in practicals also you have done it, the identifying the disease causing organism. So usually the females, this is the female which is longer in length and the males are shorter. Okay, and the tail of male is curved. Remember how to identify the male from female. The tail end is curved in case of male, whereas the tail is blunt in case of female, and it is larger in length. This is what you have to see. An example for annelida being one is nereis and the other is hirudinarea. The platyhelminthes, again in case of platyhelminthes, one more thing that you should remember is about flame cells. Flame cells are the excretory structures which maintains the osmolarity as well. Excretion and osmolarity, regulation is being done by flame cells. The Aschelminthes, they do not have any specific excretory organs. They have the pores, presence of excretory pores on their body. Okay, excretory tubes are present, excretory pores are present through which the excretion takes place. In case of Annelida, now Annelida, the locomotory structure is usually setae. Okay, in case of the annelidans. Okay, then hirudinaria, the common name for hirudinaria is leech, which acts as ectoparasite. Another very important example of annelida is earthworm. Okay, which again you should remember. Coming on to the most diverse or the phylum which is the most abundant, which is having numerous species, okay, is phylum Arthropoda. Okay, the biggest or the largest phylum in animal world, the largest phylum in animal world is Arthropoda, which is having so many different forms of species. The diversity in this phylum is very huge. Now again, if you have to speak specifically in Arthropoda, class Insecta is a major class in Arthropoda. Again, that is contributing to the diversity of Arthropoda. The examples that is mentioned here is locust, okay, gregarious pest, okay, then butterfly, scorpion, and prawn. Here, few things that you should remember. The few things that you should remember about Arthropoda is the respiratory structures, being gills, book gills, book lungs, and tracheae. Now, gills are the respiratory structures which are present in the examples of Arthropoda which are purely present in water. Means the organisms which are present in water, they are having the respiratory structures as gills. The organisms which are kind of amphibian like, which are present in water as well as on land, they have book gills as their respiratory organs. And the organisms which kind of present on land, specifically on land if they are present, they are having book lungs as their respiratory organs. And most of the insects, they are having tracheae as their respiratory structure. Okay, not only that, you also have to remember about the excretory organ. Excretory organ in case of Arthropoda is Malpighian tubules. Okay, Malpighian tubules, they have kind of well developed sensory organs. Presence of statocysts is one more very important thing here. Statocyst is nothing but it's a balancing organ. Okay, Have to remember that. Then examples of mollusca, the pila and octopus. Examples of echinodermata, asteriasis, this is what we commonly call it as starfish. Ophiuria, these are the examples of echinodermates. The adult here, the adult is radially symmetrical, whereas the larva in case of echinodermates, it is bilaterally symmetrical. You should remember this. Okay, then moving on to the hemichordata, which we see the example as balanoglossus. Now proboscis is a very important gland that we see in case of hemichordata, because this gland is responsible for excretion as well. So proboscis is a very important gland and it is seen in case of hemichordata. The examples is balanoglossus. Next, chordata, very important chordate features which we will discuss in the next slides. 5 important chordate features. Then accidia, okay, remember it as USA, okay, dollar. Now this is a trick to remember. U stands for urochordata, S stands for salpa, A stands for accidia, D stands for dolphia. Okay, these are the examples which we see in case of urochordata. Next, see very important features. You should at least remember the characters of chordates so that you can recall the non-chordate characters. Very important feature is notochord. Now before discussing this, I would like to discuss this first, because based on this we have one more very important question. The main totally there are 3 chordate classes, that's your urochordata, cephalochordata and vertebrata. These are the important different classes that we see in chordates. Okay, now apart from vertebrata there is also urochordata and cephalochordata as well, remember. Okay, now all these 3, okay, they have the presence of a notochord. Okay, division agnatha, agnatha stands for they do not have jaws, so they do not have upper and lower jaws, absence of jaws. This jaw is absent in which class? Cyclostomata. It is also devoid of scales, which we will discuss later. Agnatha stomata, they have jaws, which is divided into 2 super classes, superclass Pisces and superclass Tetrapoda. Superclass Pisces are the superclass in which we see mainly the fishes, okay, which is again further divided into cartilaginous fish and bony fish, which we refer it as Chondrichthyes and Osteichthyes. Tetrapoda is another class in which we see class Amphibia, reptiles, aves and mammals. Now notochord is present in case of chordate. It is true, yes, but all the classes of chordates, that's your urochordata, cephalochordata and vertebrata, all the three of them, they do not have a notochord or what we say spinal cord. Okay, vertebral column. Okay, we do not have vertebral column, we do not see vertebral column in all of the chordates. Okay, it's a characteristic features seen in the group of vertebrata. Okay, that's the name, that's why it is called as vertebrata. The name vertebrata came because the notochord will then at the later stages will be going to form the vertebral column. Okay, if notochord is present, they are all coming under chordates. Central nervous system is dorsal, present on the dorsal side, hollow, and it is single in case of chordates. Pharynx is perforated by gill slits. So if you see the fish here, it's a very classical example. If you see here, the gill slits are present but they are perforated. Perforated means the lines are present here. Okay, that is what we say it as a perforated or pharynx perforated with gill slits or pharyngeal gill slits. Okay, heart it is always on the ventral side. A post-anal tail, a tail is present, usually a tail is present. We may not see it in some organisms, but mainly these are the characters that we have to look for chordates. Non-chordates, the notochord is absent, CNS, if it is present, it is present on ventral side. It is solid and double. Gill slits, they are absent in case of chordates. Okay, gill slits is different, gills are different. Gills are present in non-chordates. Okay, heart, if heart is present, it is present on the dorsal side. Post anal tail is completely absent. Now you have to remember these characters. Heart, look for your own heart, it is present on the ventral side, right? The front part is ventral side, the back part is dorsal side. So if the heart, your heart, you are obviously a chordate. If your heart is present on the ventral side, here in non-chordates it will be ulta, the reverse one. It is present on the dorsal side. And your central nervous system, it is present on the dorsal side. In case of non-chordates, it is present in the ventral side. These are the small changes that you should remember. Okay, our CNS is a single CNS, whereas in case of non-chordates, presence of more than one, that is double CNS is observed. Okay, that is what we will be discussing here. Then moving on to the next part, a jawless vertebrate, petromyzon. Jawless, it jawless means obviously it is coming under agnatha, class agnatha. Then if it is coming under class agnatha, you should also remember that the class to which it belongs is cyclostomata. That is the only class that we see in case of the agnatha. Okay, a jawless example, it is not having any specific scales. If you see here, these are what we call it as scales, and these scales are absent. NCERT clearly says that they are devoid of scales. Devoid of scales means whatever these scales are there, these scales will not be present. Okay, that's why you can see that the complete body is clean. Then the other examples of cartilaginous fishes. Cartilaginous fishes is scoliodon and pristis. So if you have to remember this part, remember uh, PRISON, prison, for cartilaginous fishes. Okay, how to remember cartilaginous fishes? Obviously there are only two types of fishes. One is cartilaginous fishes and bony fishes. Remember cartilaginous fishes and you will get the bony fishes. The trick to remember cartilaginous fishes is prison. PRI stands for pristis, and at the end you see on. So in the examples where in the end the suffix, if it is there, if it is on, then that is what we call it as those are the examples for the cartilaginous fishes. Okay, scoliodon. Okay, the ending is on. Okay, remember these things. Next, so bony fishes, hippocampus, catla. Okay, these are the examples of bony fishes. The major difference between both is these have the cycloid scales, they have placoid scales. Okay, presence of the ventral mouth, their terminal mouth. Okay, and these have the bladders, the air bladder is present, and even we can see that the bony fishes are made up of bony endoskeleton, whereas cartilaginous fishes, they are made up of cartilaginous endoskeleton. Okay, then hippocampus is nothing but, it's a diagram which you see in the first part of NCERT. Okay, it is nothing but it's a seahorse. The scientific name is hippocampus. Then moving on to the next, amphibia, salamander, rana tigrina, the most popular frog in India. Okay, which will obviously see in structural organization animals. Remember that rana tigrina is the most famous frog that we see it in case of In India, mainly the Western Ghats. Okay. Reptiles. If you have to look for the reptiles, chameleon. The first is chameleon, which can do mimicry as well, mimicry and camouflaging. Crocodilus is the scientific name. Crocodile, which we call commonly. Chilon, it's tortoise. Naja is a snake. Okay. So these are the examples of reptiles. Remember, all these are having three-chambered heart, but crocodile is having four-chambered heart. So in case of crocodile, we see four-chambered heart. Rest all they are having three-chambered heart. Again, when we come to class Aves, the examples being Neophron, Strutio, Pistacula, and Pavo parrot. Remember PA parrot. I mean, sorry, peacock. Okay, peacock. Now, pista color is kind of green if you remember. So green, if it is seen, it is parrot. Okay, remember like that. Pista color is green, pista is seen. So green color animal which you see is the parrot. Okay, and pavo stands for peacock. Again, if we have to talk about aves, class aves, here it is having four-chambered heart completely. Four-chambered heart. All the organisms are having four-chambered heart. Not only that, these organisms, they are usually oviparous, which lays eggs. And one more very important point here is they are all homeotherms, means which can regulate their body temperature, or they maintain a constant body temperature. Earlier, before this class, they were all cold-blooded animals, which we call it as poikilotherms, whether it may be reptiles or amphibians. They were all cold-blooded animals. Okay, even fishes, the cartilaginous fishes and bony fishes, they were all poikilotherms, cold-blooded animals. From birds, whatever we see, they are all warm-blooded animals, homeotherms, which maintains a constant body temperature. Next, if we have to talk about mammals, okay, Ornithorhynchus, okay, kangaroo, that's Macropus, Enteropus, and Balanoptera, whale. Okay, so these are all the examples for mammals. The unique identification feature for mammals is presence of mammary glands. Mammary glands are present, and external ear pinna, the ears, whatever we see, it is present in case of mammals. In case of other classes such as your amphibians, reptiles, and aves, they do not have any specific like external ear. They can hear through the presence of a tympanum. If we see here, this represents tympanum. Okay, so this part, it's seen in case of amphibians, reptiles, as well as aves. But in case of mammals, we see the presence of external ear pinna as well. One more important point is, on top of forelimbs are modified into wings. That's one more point. And no glands are present, external glands are absent. At the base of the tail, only at the base of the tail, there is presence of some oil glands. Okay, that's one more feature of aves. Coming back to the mammals, external ear pinna, and one more very important is presence of hairs all over the body. Okay, that's one more characters of class Mammalia. Then this is summarized part. Okay, again, if we miss something, that is porifera, presence of water canal system. Okay, water canal system in case of porifera, Coelenterata presence of nidoblast cells, and Ctenophora bioluminescence, Platyhelminthes flat organs, and from this particular organism itself, from this particular phylum itself, we see diploplastic, diploplastic to triploplastic, not acoelomate, because they are still acoelomates, they are not coelomic, neither they are pseudocoelomic, they are bilaterally symmetrical, okay, and also presence of flame cells. ASCII elements, they are pseudocoelomic, annelida, they are round ones, okay, I mean the segmented body annelids, okay, three segmented group of organisms which are segmented is one is annelida, that's we see segmentation, and even arthropoda we see the body is segmented, and even the chordrates they are also segmented, okay. Now that's a very metameric segmentation is observed in annelida. We call metamerism. Mollusca, okay, again mollusca we see presence of head, the mantle part, okay, and a muscular foot. Now here we have to remember about radula, grasping organ, a very powerful grasping organ. Echinodermids we see water, vascular system, okay. Hemichordata, what happened here was Hemichordata was actually included in the chordates, but later on it was found that they are not having well developed notochord. Notochord is absent, so notochord is kind of primitively developed, therefore it was called as a stomochord. Then we talk about the chordates. We discussed about all the chordates. Moving on to the next part, that's regarding flowering plants. The basal root part, primary root, the initial formed root from radicle is called primary root. The later formed roots they are called as secondary roots. Together they form root system. Again you should remember root modifications as well, taproot modification for storage, taproot modification for support as well, okay, stilt roots, prop roots, okay, and even the roots which help for the respiration purpose, that's nematophores, okay, all those you should remember. The uppermost part forms the shoot system. The taproot, in case of taproot, the two types of roots are taproot, fibrous roots, and apart from radicle, if the roots are grown, they are called as adventitious roots. Taproot system, okay, is a type of root system in which the main primary root will elongate, will go on elongating, and it will be present till the death of the plant. So the primary root lives forever here. But in case of fibrous roots, the initial formed root, that's primary root, the primary root will die. So as the primary root dies, from the base of the stem there arises a numerous roots. So these roots are itself called as referred as fibrous roots, okay. And then the last one is adventitious roots, the roots which arise from other parts of the plant other than radicle. So apart from radicle, if the roots are arising from any part of the plant, they are called as adventitious roots. Then the regions of the root tip, okay, there are three different regions of meristematic activity, region of elongation, region of maturation. So in the region of meristematic activity, there is presence of meristematic cells. So as you know, meristematic cells, they are actively dividing cells. So the newly formed cells, they will be produced in the meristematic region, and it is covered by a thimble-like structure. Remember that it's a thimble-like structure, which we call it as a root cap. Root cap is covering the meristematic zone, and this meristematic zone is later followed by region of elongation. Here the meristematic cells will elongate in their size. Okay, there is sufficient amount of cytoplasm, which we call protoplasm, present in these cells. Then later on these cells will elongate in their size and they become large cells. Okay, then later on, still they are not differentiated, the cells are not differentiated here. So once when they reach to a specific place, specific site, that particular site, which we call it as a zone of maturation or region of maturation, is the place where those meristematic cells will actually get differentiated. Okay, differentiated means they will be either forming parenchyma, collenchyma or sclerenchyma. The cell differentiation takes place from region of maturation, and we also see the presence of root hair in the region of maturation. Okay, that's one point. And moving on to the next here, leaf, structure of leaf. This is a stem part. Now first we'll discuss this part, the leaf. This is leaf blade. Okay, this is leaf blade. The complete, this part is called as leaf lamina. Okay, this complete part is itself referred as leaf lamina. This is the midrib. Okay, midrib, from midrib the lines whatever they are arising, they are called as veins. Okay, from veins if the lines are coming, they are called as veinlets. Okay, these are veinlets. Okay, now the stalk of the leaf means the leaf is attached to one part, okay, which is getting in turn attached to the stem part. The stalk of the leaf is called as a petiole. Okay, and at the base of the petiole we see a small structure that is arising, a leaf like structure, very small structure, which we call stipule. Okay, and it is this part only wherein we will observe the axillary bud as well. So axillary bud will also come from this particular part only. Okay, which later, as mentioned here, axillary bud, which may be later on forming the the flower or maybe another branch. Okay, the branching and all, even maybe the formation of thorns, they all come from axillary bud. And the part to which the leaf is attached to the stem is leaf base. It's a very tricky and confusing question. The part of the leaf which is attached to the stem, don't go to mark petiole, because petiole is not the part of the leaf which is going to get attached to the stem. It is leaf base, the base of the leaf, this part. So this is the part of the leaf that is going to get attached to the stem, and that is what we call leaf base. Okay, two types of venation we see. One is reticulate venation. Venation means the mode of arrangement of leaf. Okay, sorry, the mode of arrangement of veins on the leaf lamina. Okay, how they are arranged? Are they arranged parallelly or are they arranged in a form of a network? Okay, if you see here, the veins, they are forming a network like structure here, and these network like structures are only called as a reticulate structure. Okay, they are referred as a reticulate. Reticulate means a network like structure. Okay, so the mode of arrangement of veins and veinlets on the leaf lamina is called as venation. Two types of venation, reticulate venation and parallel venation. In parallel venation, if you see, the veins are arranged parallelly to the midrib. So this is what we call parallel venation. In dicots, reticulate venation is observed, and in case of monocots we see parallel venation. Next, leaf. There are two types of leaf. One is simple leaf and the other is compound leaf. simple leaf which is, you know, directly attached to the stem. If this is the leaf, okay, and if this is attached and there is presence of stipule, that is what we call simple leaf. Okay, simple leaf has midrib and an entire leaf is present. Okay, we do not see the leaf incision which is touching the midrib. If the leaf is cut and if it is touching the midrib, then it is no more called as simple leaf, it is called as compound leaf, wherein the leaves appear like this. Earlier in simple leaf we used to call this part as midrib, but here we are not going to call this as midrib, but we are going to call it as a rachis. Okay, here if it is, if you see, this is what we call rachis here, because an entire leaf was present which later on incident, means forming cuts, forms the simple small leaves, leaf like structure. These are leaf like structures which we call leaflet. Don't go to call these as leaves because they are leaflets. Okay, now this is what we see. Again, there are two types: pinnately compound leaf and palmately palm. If you see the palm, it's a point at the top of rachis. If you see here, from this particular point the leaflets are coming, so therefore it is called as a palmately compound leaf. And from a single rachis, if there arises some multiple leaflets, then such type of structure is called as a compound leaf. Okay, pinnately compound leaf. And phyllotaxy. What is phyllotaxy here? The mode of arrangement of leaves on the stem. How the leaves are arranged on the stem, that is what we call phyllotaxy. Okay, phyllotaxy can be of three types. First here is alternate phyllotaxy, opposite phyllotaxy, whorled phyllotaxy. Alternate phyllotaxy refers to the arrangement of leaves wherein the leaves are arranged alternately. Okay, so if leaves are arranged alternately, then it is what we call alternate phyllotaxy. The examples that we see is China rose. In case of China rose, we see the alternate phyllotaxy. Not just in China rose, even in mustard we see the alternate phyllotaxy. Okay, mustard and even sunflower. Okay, all these three are examples for alternate phyllotaxy. Opposite phyllotaxy, guava. Okay, so how to remember alternate the examples for alternate phyllotaxy? Guava. There are two examples. One is guava, the other is calotropis. These both are examples for opposite phyllotaxy. Just remember that Calcutta and Goa, they both are opposite, right? Very far from each other, right? One comes in the northeastern part, the other comes in the south part of India, so they are opposite, right? So the example for opposite phyllotaxy is calotropis, calo Calcutta, guava Goa. So example for opposite phyllotaxy is calotropis and guava. Remember this. And whorled phyllotaxy, for whorled phyllotaxy, nerium or Allistonia is the example which you commonly see it on the roadside. Next, two types of inflorescence that we see. One is racemose inflorescence, the other is cymose inflorescence. What is inflorescence? The mode of arrangement of flowers. So how the flowers are arranged, okay, that is what we call inflorescence. The actual definition is the mode of arrangement of a cluster of flowers on the floral axis. Okay, on the floral axis, how the flowers are arranged, a single flower or maybe a cluster of flowers, many flowers, how they are arranged, that is what we call inflorescence. Then what is the difference between racemose and cymose inflorescence here? Racemose, as the name is itself suggesting, race. Race will never end. Remember how to remember the difference between the racemose inflorescence and cymose inflorescence. Race, as the name is itself suggesting, the racemose, race will never end. So that means the growth is indefinite. The growth in case of racemose is indefinite. The opposite of that is happening in cymose. So the growth is definite. Here, the end, okay, the terminal part will never end with a flower. Here it ends with a flower. And here the mode of arrangement of flowers is acropetal. What is meaning of acropetal? Always again, once again remember, if race is happening, young people will move fast, whereas the old age people will be a little behind in a race competition. So that is how you should remember. So young flowers are present at the top, the old flowers are present at the lower part, and that is what we call acropetal succession. The mode of how the flowers are arranged in racemose is the young flowers are present at the top, whereas the old flowers are present at the base. The exact opposite happens in case of cymose, wherein the old flowers, okay, the old flowers are present at the top, whereas the young flowers are present on the lower part, and that type of succession is called as a basipetal succession. Okay, and again the example to remember racemose is leguminose. You will use your legs to run, so that's how you remember. Leguminose is an example for racemose. Okay, then in case of cymose, basipetal arrangement, the example that we see here is Solanaceae. Okay, next. So how the flower is there? Okay, this was asked just okay in the NEET 2023. This question was asked, the same two diagrams were given, and it was asked to identify which type of flower it is, whether it is a hypogynous, perigynous or epigynous. Exact two images were given. You have to only look for the thalamus. This part is called as thalamus. Only look for this thalamus and this ovary. Okay, now thalamus is present here, ovary is present on top of the thalamus. So such type of a flower type is a hypogynous. I usually say this, right? How do you define hypogynous flower? Ovary is superior, that's why flower is hypogynous. Never say like this. Okay, the ovary is situated on top of thalamus, and all the other floral parts, they arise below the ovary. That is why the flower is hypogynous. You can say that the flower is kind of inferior. What is the position of ovary? Ovary is superior. When the question is asked with respect to position of ovary, the ovary is superior. Okay, then here if you see, the ovary is still present here, the thalamus is there here, okay, but it has not enclosed, it is not covering the ovary. And if you see here, ovary is present kind of at the same level. Okay, I don't know why they have given the diagram like this. It should be here somewhere, but they have mentioned it there. So in both B and C, remember that the level of ovary and the thalamus is somewhat at same level. Okay, you see the thalamus is half covering the ovary, so therefore this condition is said to be, the flower is said to be perigynous. And the third situation, the third scenario is what we call epigynous. If you see here, the ovary is completely inside the thalamus. The thalamus is here. The thalamus is opening here. The ovary is below the thalamus or inside the thalamus. So such type of the flower is referred as or is called as epigynous. Here the ovary is inferior. Here the ovary is superior. and here the ovary is semi-inferior in case of perigynous. Okay. Now here, if we have to remember the few examples here, the hypogynous situation is observed in most of the flowers, okay, wherein the ovary is situated above the thalamus. We observe this condition in almost all the families, the Solanaceae, okay, the Asteraceae, Malvaceae, okay. In all those examples, we find hypogynous. Don't just consider the examples that is prescribed in your NCERT, only that, because as we are discussing morphology of flowering plants, there are 5 additional families as well. So think from that point of view as well. Okay. Epigynous, if you see the florets of Asteraceae family, okay, the ray floret, okay, shows epigynous condition. Okay. And with respect to perigynous, you have to see about the peach and plum. Okay. And even the rose. Now parts of a flower: pedicel, it's a stalk of the flower; calyx, which protects the flower in bud condition; corolla, which is the most colorful and bright part of the flower, which is responsible in attracting the pollinators; gynoecium, the female part of the flower; androecium, the male part of the flower. Okay. Then estivation. What is estivation? The mode of arrangement of the sepals and petals within the floral bud is called as estivation. So if you observe the first one, here is valvate estivation. What happens in valvate estivation? The sepals or petals, they are almost in contact with one another, but they are not overlapping. So such type of arrangement is called as valvate estivation. And in the next imbricate estivation, twisted estivation, what happens here is the sepals or petals, they are overlapping in a particular manner. The pattern of overlapping is very specific. Now if we see, one part of the sepal is present inside, the other is outside. Again present on inner part, outer part. So again one part is present internally and it is going outside. Again one part of the petal is inside, the other is outside. So that is what we call twisted estivation. Okay. And the next being imbricate estivation. Imbricate estivation is a type of estivation in which the overlapping is irregular. There is no regular overlapping. Okay. If you see, you cannot come to a conclusion here. The one part is present outside, the other is inside. You are expecting it to be the same, but here again it is outside, again the another part is again outside. Here this is inside, this part is outside. These two, both the parts are present internally only. So the pattern of overlapping here is not regular. Okay. When you come to the last type, that is vexillary estivation. What is vexillary estivation here? We see one large petal which we call standard petal. Then these two are small petals which we call it as wing petal. Okay. And the smaller two ones, they are called as keel petals. Okay. These both are fused, and this is very important because vexillary estivation is observed in pea plant. Okay. Remember this part. Okay. Next, the placentation. What is placentation? And the mode of arrangement of ovules inside the ovary with the help of placenta, how it is arranged, so that is what we call placentation. Again, going for the first one is marginal placentation, second one is axile placentation, third one being parietal, fourth free central, fifth basal placentation. Example for marginal placentation is pea plant. Axile, it's seen in most of your Solanaceae family members. Not only in just Solanaceae family members, even in Malvaceae family members, we see the axile placentation. Even hibiscus, which you commonly have performed the practical experiment wherein you have taken a section of ovary and observed pentalocular structure, right? So that's how it is similar to this one. So the examples for exile placentation being hibiscus and all the Solanaceae family members. Remember that, even citrus as well. Then moving on to the next one, the which one? Parietal one. Parietal one here refers to the mustard and argemone. Okay, argemone and mustard are examples for parietal placentation. And in case of free central, prime rose and dianthus. Dianthus and prime rose are examples for free central. Basal placentation, all the sunflower, most of the Poaceae family members, they show or they exhibit basal placentation, because if you observe the one character of wind pollinating flowers, okay, they usually don't they usually have only one ovule present inside the ovary, and it shows basal placentation. And in general, if you have to say sunflower, marigold, they are all coming under Asteraceae family members. So all the Asteraceae family members, they exhibit basal placentation. Then parts of a fruit, outermost covering is called epicarp, inner endocarp, then middle is mesocarp, inner is endocarp. The seed is present inside that. So the outer thin part, if you have consumed a mango, the outermost thin part is called as a epicarp. The part which you consume, amji here, okay, amji that you consume here, what happens here is the part which you consume like anything, okay, till you reach a part which is hard and stony, that hard and stony part is called as endocarp. Okay, this is a hard and stony. If it was edible, you people would have not left that also. So the endocarp is not edible, but mesocarp is edible, and epicarp is a thin fleshy part that is present externally. In case of mango, it's an example for drupe. Type of fruit is a drupe. Okay, drupe, both coconut and mango. The outermost part is here referred as the fibrous roots that we see here, and inner mesocarp. Okay, where is epicarp means, when you see the coconut on a tree, the green color part, that is epicarp actually. Okay, then the innermost part here is the fibrous part, here is mesocarp. I mean, the inner part is referred as the endocarp. Next, structure of dicotyledonous seed. Dicots, presence of two cotyledon. At the top there is presence of plumule, and at the base there is radicle. So hilum, if you remember the second year structure, okay, the oval part, okay, here there is embryo sac, the nucellus, outer integument, inner integument, the micropyle part, funicle. This point, the point where the ovule fuses with the funicle, is called hilum. So that hilum part is present here, and we see the micropyle part as well. The part which is not covered by any integument is micropyle. Then in case of monocot, you have to remember aileron layer. Aileron, it is a proteinaceous layer made up of protein. Again, in cell the unit of life, you will come across types of plastids. There are a total of three plastids: chloroplast, chromoplast, and leucoplast. Leucoplast are colorless plastids, whereas chromoplast are colored plastids. Chloroplast is specifically the plastid which is having chlorophyll present in them. If it is leucoplast, leucoplast has to do something. It is not having any color, so it is not doing any work. So it will carry out the process of storing of food materials based on what it stores. If it is storing carbohydrates, it is called as amyloplast. If it is storing protein, it is called as eleroplast. If it is storing fats and oils, it is called as elioplast. Okay. Then coming back, this is a floral formula for the Brassicaceae family, which is mentioned in your textbook. The anthers, they are arranged in a tetradynamous condition. Okay. This is tetradidamus. For didydamus, there is salvia, not salvinia, which we discuss in pteridophytes. Salvia is an example for didydamus condition, whereas for tetradidamus, the condition is observed in mustard family, that's Brassicaceae family. Clear? Then, moving for the next one, this is Solanum nigrum. The most ignored part here is this part. They are considering the standard example for Solanaceae family here is Solanum nigrum. Okay, most of you will not observe this part. Makoi belongs to which plant family? If the question comes, Makoi is the common name. The scientific name is Solanum nigrum. Okay, and remember Solanum nigrum is having one more name, that's nightshade plant. Okay, so if it is asked as nightshade or Makoi, the scientific name being Solanum nigrum, it belongs to which family? Solanaceae family only. Okay, and one more very important point that you should observe here is the gynoecium, the arrangement of gynoecium. If you see here, it is not exactly straight like this. It's not like this. It is slightly tilted. Now this tilted part is itself referred as obliquely arranged gynoecium. Okay, so you have to remember this part as well. Next, moving on to the anatomy of flowering plants, the two types of stomata that we observe, one is the kidney shaped stomata and dumbbell shaped stomata. The kidney shaped stomata is found in dicots. Remember this one. Okay, it's very important. The guard cells are kidney shaped, and one question that has appeared, the thin name the component of the stomatal apparatus whose outer side is thin. Once again I will tell, understand this very carefully. Name the part of the stomatal apparatus whose outer side is thin but internally it is thick. That is not, don't go to mark stomatal pore. Stomatal pore, if this is the stomatal pore, the outer side it will be thick only, right? So it's not the stomatal pore. The correct answer is guard cells. So these are guard cells. If you see externally on the outer side, it is very thin, but internally it is completely thick. So therefore, the part of the stomatal apparatus which is thick externally, internally, if I mean thin externally, internally if it is thick, it is guard cell. Okay, and can be open and closed. Presence of the chloroplast as well. Now here it is a dumbbell shaped stomata. Okay, in monocots, observed in monocots. Here the kidney shaped stomata is observed in dicots. Next, if we have to look for arrangement of vascular bundles, remember these three R's. Okay, what is this 3R? The radial arrangement. What is radial arrangement? Here the xylem and phloem, they are arranged radially at a different radius. So if you consider this point as the center, okay, from this point the xylem is present here, but the phloem is present here. Xylem is present here, phloem is present here. So the vascular bundles are present radially at different radius, but where they are present? In roots. So 3 R's. 1 R stands for radial arrangement at different radii in roots. So that is 3R trick to remember, okay, where radial arrangement is taking place. Next, if we have to speak about the next part, that's conjoint arrangement, conjoint open and conjoint closed. This is seen in case of stem part, usually seen in stem. Okay, what happens here is the vascular bundles, they are arranged togetherly. Phloem is present here, xylem is present here. So internally the xylem is present, externally phloem is present. In between them, if there is no layer, then such a type of arrangement is called as a closed conjoint. Okay, it is seen in monocots. And here if we see, in between phloem and xylem, there is presence of cambium. Cambium is essential for secondary growth of the plant. Okay, so this will be referred as conjoint and open type. Conjoint and open. How to remember? Whenever you open door, if you observe cambium, if you open the door, you will observe cambium. So that's how you should remember. So if it is open type, you will observe cambium. What if you close? If you close the door, the cambium is absent. Okay, दरवाजा अगर बंद करोगे तो you will not find cambium. If the door is open, you will find cambium. So open type of conjoint is present in dicots. Now it is very essential for secondary growth, remember that. And secondary growth is usually seen in dicots and gymnosperms, but in case of monocots, secondary growth is absent. Why secondary growth is absent? Because they do not have cambium present in them. Now moving to the next part, TS of dicot root and TS of monocot root. The major difference between these two is presence of the vascular bundles. In case of dicot root, there is presence of only 4 vascular bundles here, which is arranged again radially because we are talking about roots. So in roots, the vascular bundles are radially arranged. In case of the monocot, here we see more than 4. Okay, we usually see the condition called as polyarch. Polyarch in case of monocot and tetrarch in case of dicots. That's how we differentiate between the monocot root and dicot root. Mainly we differentiate based on that. Okay. Then here one more thing is, if you remember in root, the hypodermis is absent in case of roots, but hypodermis is present in case of stem. So in case of dicot stem, the hypodermis that is present here, okay, this hypodermis part is made up of collenchyma. What is hypodermis? Below the epidermis, the portion is referred as hypodermis. This hypodermis is made up of collenchyma in dicot stem. Okay, and you see the presence of pith in the early stages. In the later stages, the pith gets reduced because of secondary growth. Okay, now if you see, this is what we have seen earlier. On the inner side there is xylem, on the outer side there is phloem. In between xylem and phloem there is cambium. So this part is cambium, on the upper side there is phloem, and here there is presence of xylem. In between phloem and xylem there is presence of cambium. Okay, the later formed xylem is called as metaxylem. The early first formed xylem is called as protoxylem. This is completely different in monocot stem. In monocot stem, the hypodermis is made up of sclerenchyma. In case of monocots, the hypodermis is made up of sclerenchyma, and here the ground tissue is further not differentiated. The cortex region is not again further differentiated into endodermis, pericycle, the vascular bundles, the conjunctive tissue, the pith. The ground tissue is not divided like that. Okay, so overall, the complete tissue here that we see here, ground tissue that we see here, is having vascular bundles. The small sized vascular bundles are arranged in the peripheral side, whereas the developed vascular bundles, the large sized vascular bundles, they are present exactly in the center part. Okay, that's how we differentiate monocot root from dicot root. Here we will use the word scattered. The vascular bundles are scattered throughout the ground tissue, wherein the smaller vascular bundles are present peripheral side, whereas the large sized vascular bundles, they are present near to the pith part internally. Next, we will discuss about the tiers of leaf. Okay, leaf. The uppermost epidermis is referred as adaxial, adaxial epidermis. The lowermost epidermis is called as abaxial epidermis. Now, it is also called as a dorsal part, and this is referred as ventral part. Now, how to remember these name, or again this can also be called as upper epidermis and lower epidermis. Adaxial has a D in it, so D for dorsal. Abaxial doesn't have any D in it, so you can remember adaxial and dorsal, abaxial ventral side. So in case of dicot stem, dicot leaf, sorry, in case of dicot leaf, the upper epidermis is not having any stomata. It has stomata only in the lower part of the epidermis. Now, that is the reason why it is called as a dorsiventral leaf. What it is called as? Dorsiventral leaf. Now, why it is called as dorsiventral leaf? Because on the dorsal side, there is no stomata, whereas on the ventral side we see stomata. That's one reason. And the other reason is the next part that is present after epidermis, either from upper epidermis or lower epidermis, it is called as a mesophyll cell, mesophyll region. Now that mesophyll part is further divided into two types of mesophyll cells. One is palisade and the other is spongy. The air cavity is present in spongy mesophills. The palisade parenchyma is not having any air gaps. Okay, so the substomatal region is present wherein the air enters into the cells, diffusion occurs, wherein the oxygen and the gases gets exchanged here, and the vascular bundle is present here. When we talk about monocot, okay, both upper and lower epidermis has the stomata. Okay, the stomata is equally distributed. Not only that, the mesophyll cells here, they are not differentiated into palisade and spongy. Okay, here we just refer it as mesophyll cells. Presence of vascular bundle. Not only that, there's one important point that we should discuss here is about bulliform cells. Bulliform cells are the large empty colorless cells that are usually present in the grasses. Okay, and they are responsible for curling of leaf to reduce water loss, to reduce transpiration. What happens? The bulliform cells are present, which rolls itself, curls inwards, not outwards, curls inwards, so that the the stomata gets covered, so that when sunlight falls on it, the evaporation or the transpiration process should not take place. So for that, bulliform cells are present in monocots. Next, we'll move on to the seventh chapter, that is structural organization animals. We are talking about frog. Forelimb is having 4 digits, hind limb is having 5 digits. In case of male, there is presence of copulatory pad in the forelimb, okay, and also it has vocal sac, okay. Vocal sac and copulatory pad are the characteristic features of a male frog, okay, and forelimb is not webbed whereas hind limb is webbed. Because of presence of webbed feet, it is going to help for swimming, okay. Not only that, eye has one membrane called as a nictitating membrane, which covers the eyes when present inside the water. The body is divisible into head and trunk part, okay. The tail is absent. Tail is present only in larval stage but not in adult frog, okay. Not only that, you have to remember other things as well. This is tympanum through which the frog hears, okay. Apart from that, you should always remember that the respiration in case of frog occurs by three means. Whenever the larval stage is there, okay, it respires through gills during larval stage, okay. Then when it grows into an adult, it can either respire by the buccal cavity, okay, which we call buccopharynx, and it can also respire with the help of skin as well, okay, cutaneous respiration, okay, and even presence of the lungs, okay, it can respire through lungs, buccal cavity togetherly. Then it can also respire through skin. If it is respiring through lungs, we call it as pulmonary respiration. If it is respiring through the skin, it is called as cutaneous respiration, okay. That's about the general features of frog, okay, which you should remember. Now moving on to the digestive system. The digestive system, mouth is present, which is a bilobed tongue, which is having bilobed tongue, which when engulfs any insect, okay, it directly enters into esophagus. But esophagus is very short. Esophagus, the intestine is very short. Why it is very short? Because it is a carnivore. Because it is carnivore, and it is a very small primitive animal, it requires more amount of oxygen to break down that. But as it is a polyculture, it has to consume its energy in breaking down all the components and for the movement of food. So the carnivore is very reduced. I mean the intestine is very reduced because it's a carnivore, okay. Then the food enters through the mouth, passes to the pharynx part, esophagus, then it reaches stomach, then it moves to digest intestine. Here there is presence of duodenum and ileum, okay, and followed by rectum. The undigested food particles enters through rectum, and then the food comes out in through cloacal aperture. Remember that the partially digested food is called as chyme, and the chyme is formed in the stomach part where there is presence of gastric juices and gastric enzymes and HCl. So because of the action of gastric juice and the HCl, the food becomes partially digested, which we call it as chyme, which later enters into intestine, okay. It is the intestine part where the complete digestion takes place because of the action of gallbladder, which secretes the bile juice as well as the juice that is secreted from the liver, okay. So the digestion takes place in the intestine. The undigested food passes through comes to the rectum. This is the rectum. From the rectum it reaches cloaca, and from cloacal aperture it the undigested food is released out from the body, okay. Now the next part, just a second. I guess I pressed the delete option by mistake. Just give me a moment. Yeah. So the next part is male reproductive system. Male reproductive system. Here you should remember that the reproductive structures are testes. Testis is not just attached to the kidneys. Okay, it is not just a structural connection, but whatever the testis are going to produce the sperms, those sperms they are going to enter directly into the kidney. Okay, it passes through the Bidder's canal and it reaches the kidneys. That's very important. So the testes here they are structurally as well as functionally connected. Structurally and functionally they are connected to the kidneys. Okay, remember, structural as well as functional connection. Functionally they are connected to the kidneys. Now these sperms enter into the kidney by the help of vasa efferentia, which may be 10 to 12 in number, similar to that of in case of human beings. So vasa efferentia is going to carry the sperms and enter into the kidney where there is presence of Bidder's canal and then L-shaped tubule, which is then going to enter into the ureter. Now if you see here, this ureter, it is going to carry from kidney, it is going to carry the urine, but the as testis is attached to the kidney, even this ureter carries the gametes as well, which is sperms. So this ureter is also referred as urinogenital duct. Urino because it carries urine, genital because it is going to carry the sperms. Therefore ureter in case of males. Okay, so once again if you have to see, the ureter in case of males is called as urinogenital duct because from kidney it is going to receive the urine. At the same time, even the testis are structurally and functionally attached to the kidneys, because of which the sperms will also enter into the kidney only. So as the kidney is connected to the ureter in males is called as a urinogenital duct. Okay, and then this urinogenital duct is going to open into cloaca. Cloaca say cloacal aperture where they are being released. Okay, if we have to talk about the female reproductive system in case of frog, the ovary, the female reproductive structures, they are attached to the kidneys. Structurally they are attached, but functionally they are not attached. The ovary are going to release the eggs. Those released eggs, 2500 to 3000 eggs, they are going to be released, and these released eggs they are going to be collected in a separate egg carrying duct which we call oviduct. So from oviduct it is going to enter, it is going to pass through and then released into the cloaca. The kidney is going to carry the urine in the ureter, and the ova or the eggs they are being carried from the oviduct. This is what we call body cavity. Remember, the khali space here is called as cavity. So the released eggs they are going to enter into the oviduct and ultimately passes through the oviduct and reach cloaca, and they are going to open into cloaca. So a question that can be asked from the cloaca part is, the cloaca is a common aperture or opening for urine, undigested food that comes from rectum, and also the gametes. So that's a very important point to be discussed here. Now the arrangement of the peritoneum layer here, based on that how the testis is attached to the kidneys, it is called as mesorchium. Here it is referred as mesorchium. The method of how the testis is present on the kidney is called as mesorchium, and in case of female it is referred as mesovarium. Okay, so next is cell the unit of life, which we will continue at 3:30 class. So we'll complete this part and then we'll go on to the completing the next remaining topics from the second year. Okay.